Welcome everyone to another video in Biology 2, and this time around we'll be talking about another important topic, and we start off the discussion by asking this question. Do all cells in our body have the same DNA? Do all cells of our body have the same blueprint called DNA? Well, the answer to this is you already know. Generally speaking, yes, our cells have the same DNA. And that will lead us to another question, and that question would be if all our cells in the body would have the same DNA, would have the same blueprint, then why do they look different from each other? Why do eye cells look different from liver cells and liver cells look different from muscle cells? And the answer to that is of differences in gene expression. So it's a good thing that there is differences in gene expression because the eye cell will look different because they have a different set of genes active at the same time different set of genes that are deactivated or silent as compared to the liver cell that will also have a different set of genes that are active and deactivated at the same time. So this will result to differences of the eye cells, the liver cells, and all of the cells in our body. And it's a good thing that the gene expression will be different, otherwise our body will experience chaos. And the reason for that is explained by the video of the Amoeba Sisters, where in one part of it, it will be showing that the eye cells, what if they made a mistake and instead of just activating genes for the development of the eyes, they were able to activate development of genes for the stomach. And what will be the problem? As you can see here, the sensation will become unpleasant because if the eye cells now activate genes for the stomach cells, it might be producing hydrochloric acid, leading to very unpleasant sensation, as you already probably would have expected. Now, with that, our topic for today is controlling genes, or module 2.6, which is control of gene expression, or regulation of gene expression. It's a good thing that our cells are able to regulate its expression, leading to different parts of our body and harmony in our body. So our objectives for module 2.6 is number one, to compare and contrast the regulation of gene expression in prokaryotes. And take note, we will only be talking about prokaryotes today because eukaryotic gene expression will be a little bit more complicated, that will be addressed in your higher biology or sometime later. Number two, our another objective is for us to identify at which stage of gene expression would control occur. And with that, let's have a little concept check, a little review. What is a gene? Because it's very important because regulation will be happening at the gene. So the gene is described as the nucleic acid sequence necessary for the synthesis of a functional gene product, and this will remind us of the central dogma of molecular biology, wherein the gene, which is a segment of DNA, produces the RNA, and the RNA produces the polypeptide. So just remember, the gene is a nucleic acid sequence, a DNA sequence necessary for the production of a gene product. And the reason for this, we are studying this all over again, is because the control of gene expression will start with the gene itself. So let's now talk about gene expression control that happens in the prokaryotes, and you have to be introduced to what we call as the operon. What is the operon? The operon is defined as a group of genes that are transcribed together to produce a single messenger RNA molecule that would be coding for multiple proteins. So take note of the important parts of this definition. Number one is this is a group of genes. That is why prokaryotic DNA or genes or prokaryotes are said to be monocystronic because polycystronic, because they would be producing a single mRNA as mentioned here, but they will be coding for or producing multiple proteins as compared to eukaryotes that are termed to be monocystronic because their mRNA would be producing only one kind of polypeptide. Now take note that the operon has a number of structures that are very important. First of all, this would be the promoter, the promoter will be containing or will be the section of DNA, a gene, that would be where the RNA polymerase will be attaching itself to start off transcription. The operator gene will be very important because this is where the repressor protein will be attaching itself to prevent transcription. We'll be dealing with that a little bit later. And another part of the operon will be the structural genes, and these are the DNA sequence or segments that will be later on producing the actual proteins, the actual polypeptides that are needed by the cell. Now take note that upstream there is what we call as a regulatory gene, and the regulatory gene will be producing either an activator or a repressor, which will be very, very important to the kind of gene regulation that will happen to the operon. Take note that the regulatory gene has its own promoter, so it's separate from what we call as the operon. So let's continue on discussing. What are the two kinds of activator? While the negative gene regulation will have what we call as a repressor. So take note of this one, the negative gene regulation, the binding of the transcription factor would be present in the promoter for the positive gene regulation. So in summary, positive gene regulation, genes are expressed because of the presence of the activator, which are transcription factors on the promoter gene. Negative gene regulation, on the other hand, the genes are not expressed because of the presence of the repressor proteins that are bound to the operator gene. So let's now look at the kinds of negative gene regulation. We are now done with comparing the positive and the negative gene regulation, and we'll be going back to the positive gene regulation a little bit later when we'll look at the lactose operon. But let's now take note of two kinds of negative gene regulation, and there are two of those, letter I and letter R. And what are those? The first one is called inducible gene regulation. The other one is repressible gene regulation. As you can see by the name itself, when we say the word inducible, it means that it is originally turned off, but it can be induced to be turned on. On the other hand, when we hear the word repressible or the word repressed, it is originally turned on, that it can be repressed to be turned off. And that would be the major difference between the two kinds of negative gene regulation. So let's dive into the details of this, the inducible and the repressible. So in this, we can see here that the lac operon would be representing the negative inducible operon, while the tryptophan operon would be representing the negative repressible operon. Looking at the diagram, we have here the summary that the inducible operon, in the presence of the metabolite, it will be able to turn it on. Originally, it is turned off, but the effect of the metabolite is to turn it on. And that metabolite is called lactose. For the repressible operon, the presence of the metabolite will make or has the effect of the originally turned on operon to be turned off. And the metabolite here will be the tryptophan